Welcome back, children. Now we want to know an idea regarding cell. Okay, why are cells so small? Okay, we want to see why the cells are not enlarging in their size. Why it is so small? Okay, the reason is they need to survive in a particular organ or particular area. Means they need to interact with their surrounding environment, and they need to interact means only certain level they can able to grow. If it will grow beyond that size or beyond their volume means automatically that is abnormal growth. There we can see cancer cells. So if it is a normal cell means they need to interact with the neighboring environment and they should have one certain growth only. That's why it is very smaller in size. And if the cells grow beyond a certain limit, enough materials will not be able to get it from the neighboring cells, because everything was assigned one limit only. And when it is crossed only we can see abnormal. So normal cells means they need to get some material or food material from the neighboring cells, and they can able to grow only their cell volume, or they can able to maintain their cell volume or their cell size. Actually here normally we have learned in the animal kingdom chapter regarding coelomate. Coelomate means they'll have some body cavity. With that, organs can able to enlarge in size. Enlarge in size means only the number of cells will get increased, not the cell size. Not the cell size will increase. So that is only you want to remember. Increases in the number of cells, not in the volume. Is that clear, children? And here that's why the cells are so small. And surface area in the area of the outside of the cell is called plasma membrane, and the volume is how much space is inside the cell. So that protoplasm is covered by plasma membrane. With that only they can able to extend, because they have a one boundary wall like plasma membrane. So again it will enlarge means the plasma membrane will tear, that's all, and every component will come out. So to certain extent only the cell can increase. And here the ratio is the surface area divided by the volume. If the surface area to volume ratio is small, the cell is very big. So here you want to remember, surface area to volume ratio is small and the cell is very big. So the reason is volume and the surface area. Already the surface area is large, and automatically the surface area will be filled with volume and the cell size will be bigger, as like we have learned in ostrich egg, no? So there the surface area also bigger, and the volume can be inside the surface area is also increased. So we can see bigger size cells. And when this happens, the cell must divide into smaller cells with favorable surface area or volume ratios. So here what you want to remember, why are cells so small? Because already it was designed, the surface area only limited area, and to that only the volume will be filled. And it varies from some of the cells. Why the cells are bigger in sizes in some exceptional alone, there the surface area is bigger and the volume can be filled in that. So the cell size is bigger. So that is only you want to remember. And come to cell shape. So cell shape we have learned, it is not a constant shape. Every cells will have a constant shape. We can't tell that. So cell shape may be spherical like a round, or polygonal, okay, or a disc-like structure, okay, or cuboidal structure, and or a columnar like a epithelial cells and all, like a columnar cells only, or a spindle shaped, okay, like muscle cells and all, spindle shape, or a fiber, a lengthy fiber like structure. So this is only we call it as a different shape. So spherical means round, polygonal, spherical means round, polygonal we know, and disc like, and cuboidal, the structure of cuboidal, columnar, okay, like a lengthy epithelial cells and all, we can see like a columnar, like a pillar like structures, okay, and spindle shaped as in the case of muscles, and fiber, very lengthy cells, that is only we call it as fiber. So the structures will be different. And generally cells are spherical, but in multicellular forms due to pressure it will become a polyhedral. Normally while forming it will be like a spherical only, children, but because neighboring cells they all will give pressure, so in that area they need to adjust, means the shape soon was changed into polyhedral. In plants as well as we can see, cell wall is appear, and that's why it will attain a common structure like hexagonal or polygonal. And cell sometimes changes shape because of functions as like amoeba and leukocytes. So we can call it as what children? So like amoeba and leukocytes, constantly they will change their shape. So this is, we can call it as a pleomorphism. They will change their morphology. Amoeba and leukocytes. What is leukocytes? Your white blood cells. So depends upon their function, they'll change their shape and size. So we'll tell that this, they'll contains pleomorphism, change their structure. And even animal cells, they'll have an irregular shape, and plants are typically rectangular or cube shaped because of the presence of cell wall in it. I think you have understood regarding the shell shape. And these are some common example, children. Now look at the Pinularia, Chlamydomonas mural oval, Paramecium. We have learned a lady shoe slipper. Acetopularia, I have told, very lengthiest but single cell, children. This is very lengthy, but it is a single cell only. And come to Escherichia coli, Caulerpa. This is again algae only. And onion peel cells, we can able to see these kind of structures. We did some practical in grade 9. So onion peel cells. And red blood cell, this is a biconcave structure. And epithelial cell, this is a columnar, I have told, this is a disc shaped structure or concave like epithelial columnar like structure. And muscle shape, this is spindle shell. And nerve cells, it is having like a dendritic structure. They'll have a elongated body. And amoeba. So now look at this, all are single cell only, children. All this thing are single cell, but they'll vary in their shape and size. And come to types of cells. We have learned in the lower classes two category only, okay, prokaryotes and eukaryotes. And Daughtry, okay, in the year of 1957, Daughtry in the year of 1957 classified cells into prokaryotes, meaning we know eukaryotes true nucleus. On the basis of their structural organization of the nucleus, this person, Daughtry, 1957, he has classified. And we know the definition for prokaryote and eukaryote, no children. We have many of time I have explained that. So we don't want to go in detail again with the prokaryotes and eukaryotes. You simply want to remember, not well developed nucleus, prokaryotes, well developed nucleus with a nuclear membrane, we can call it as eukaryotes. And Dodge et al. 1966 proposed that this is very important in competitive based question, children. Okay, Dodge et al. 1966, he has proposed a third type of category, that is Mesokaryotic, and which can be placed between prokaryotes and eukaryotes. What is the mesokaryotic example? Dinoflagellate. Okay, dinoflagellate. Why it was kept between pro and eukaryote? Prokaryotic means there is no histone proteins. Eukaryotic means histone proteins will be seen. But mesokaryotic, like dinoflagellate, where nucleus is covered by a nuclear membrane, but there is no histone protein. That's why we can call it as a separate group, mesokaryotic. Is that clear, children? Next, we will go quickly with the prokaryotic cell and eukaryotic cells, okay, and mesokaryotic cells. So, prokaryotic cells I have explained already regarding the bacterial structure while taking the biological classification. And here what you want to remember, a concept, first concept is, it is a very primitive type of nucleus where the nucleus is not at all bounded by a nuclear membrane. That is first. So we would call the nucleus as nucleoid. We would call the nucleus as nucleoid in bacteria. And there is no membrane bound cell organelles like mitochondria, chloroplast, and we can see the plasma membrane, it will fold. I have shown this plasma membrane. They will fold in some certain region, that is only we can call it as mesosomes. This also I have explained while taking the biological classification. And the nuclear material is lies diffused in the central part of the cells. It contains DNA, but it is not associated with what protein? Histone protein. Histone protein is absent in prokaryotes. And prokaryotic cells are smaller than the eukaryotic cells that we know. What is the common example? Bacteria, archaebacteria, blue-green algae, mycoplasma without a cell wall of bacteria, and rickettsia are included in this type. So this is a bacteria, flagella can be seen, and we can see a big flagella remaining around cilia, and this is we can call it as only ribosomes can be seen in bacteria, and plasma membrane can be seen, and capsid. This cell wall is covered by a capsid, and we can see the cell wall. So we can see cytoplasm, ribosomes, and nucleoid, and cytoplasm is covered by plasma membrane. Plasma membrane is covered by a cell wall. Cell wall is covered by a capsid. And we can see pili, P-I-L-L-I. It is meant for conjugation of neighboring bacteria. It is meant for reproduction. Next, children, will see the eukaryotic cells. We all will know eukaryote means true nucleus. So we can see a true nucleus, nucleolus, and we can see all the cell organelles, mitochondria, endoplasmic reticulum, Golgi body, plastids, everything. And we can see a histone protein in it. And what kind of groups we can see? Plants, animals, every groups we can see here. Plant group except blue-green algae. Next, we'll see mesokaryotic cells. As I explained, example for mesokaryotic cell is a dinoflagellate, and there the genetic material is surrounded by a nuclear membrane. But what is absent here, children? Absent is no histone protein. No histone protein. That's why they have kept between a prokaryotes and a eukaryotes. So no histone protein, and nucleus is very larger only. And this mesokaryon was introduced by Dodge. Okay, Dodge et al. Only in 1966 they have given this new category, mesokaryotes. And these types of cells which are intermediate between prokaryotes and eukaryotic cells, that's why we can call it as mesokaryotic cells. And here we can see mitotic spindle also not formed. What do you mean by mitotic spindle means? I will take in this unit even regarding cell division. There I will give a detailed structure regarding what is spindle. Spindle means it is just like a fiber-like substance which is very important during cell division to separate chromosomes into two different cells. And what is the common algae for example is gymnodium and peridinium are the common example for this group. Is that clear, children? So this is mesokaryotic cells. Apart from that, there are some exceptional like Mycoplasma, no? That is PPLO, pleuropneumonia-like organisms, already have explained. And also some size, no, it is a very lower size. Mycoplasma is, though it comes under prokaryotic, but it is very lower than the prokaryotic. Is that clear, children? And also other group, no, like a simplest prokaryotic, the same like PPLO only you want to remember here. So you remember only this, children, Mycoplasma, PPLO and their size. It is smaller than the prokaryotic. That is only you want to remember here. And who placed PPLO with bacteria means this. Okay, Litterson only. He has placed PPLO with the this group, bacteria group. So this is the structure of BBLO, children. I have told, no, pleuropneumonia-like organism, smaller than the bacteria, but it is comes under bacteria because it has the ribosomes and other cell organelles are absent. And we can see nuclear material is very free. So that's the thing they have kept under prokaryotes. And here, what is the very important? Absence of cell wall and mesosomes. That is very important, children. We are telling characteristic of bacteria is what? Characteristic of bacteria is presence of cell wall and mesosome. The plasma membrane is folded and they will form a structure called mesosome. And both also absent in the mycoplasma. That only you want to remember under PPLO structure. And the difference between prokaryotic and eukaryotic cells. Okay, that already we have learned from the lower classes, no? Just difference alone we can see quickly. And compartments, prokaryotic no, eukaryotic we can see different compartments. Okay, like mitochondria, like all those things, compartments we can see. And cell wall is present in prokaryotes, but in eukaryotes cell wall is absent. Only in plant cells we can see. Plasma membrane is present, in both also, that is lipid bilayer, and cell organelle is absent in prokaryotes, and we can see only ribosomes, and eukaryotic cells we can see all the cell organelles, and we can see the plasma membrane also, but there is no mesosomes. And respiratory was carried out by in prokaryotic is through mesosomes only. That is that it is a part of plasma membrane. And respiration in eukaryotic is mitochondria, and capsule is present surrounded by the cell wall, and capsule is absent in the eukaryotic. And what is the photosynthetic apparatus here? And here like a pigments only can be seen, and it is not a membrane, like no plastid, but pigments can be seen in the blue green algae. But here like a chloroplast, a separate organelle will be there. And clear nucleus is absent, so nucleus is not distinct, so we can call it as nucleoid, but here we can see distinct nucleus. And other nuclear membrane is absent, here present nucleoplasm, it is undifferentiated from cytoplasm because there is no separate nuclear membrane, no? So that's the thing we are called as undifferentiated from cytoplasm, but here it was differentiated. And hereditary material is DNA or RNA, but DNA will be circular in shape in bacteria, and it is not linked with a histone protein. But here we can see strands of DNA, like it's single stranded DNA or double stranded DNA, and it is joined with the histone protein. And nucleolus is absent, here nucleolus is present. What is the role of nucleolus? It is a factory for ribosome. Nucleolus role is factory for ribosomes. And flagellum is absent. Children, this is very, very important. And it is composed of 9+2 pattern. And here 9+2, but we can see tubulin protein. This I will explain while taking in the flagellin protein. Cell organelle I will take. That time I will explain the condition of what is 9+2 and what is 9+2 tubulin. And cell division is amitosis. That is simple binary fission only. And here we can see mitosis and meiosis. And ribosome present in both prokaryotes and eukaryotes. But here the ribosome is 70 years. That is 2 subunits can be seen, 30 years and 50 years. But here 80 years, that is 40 years and 60 years. So not 40 plus 60 not 100, that is 80. And here 30 plus 50 not 80, it is 70. What does S denotes? S denotes for Swartberg unit. So 2 units joined together in ribosomes. Here also 2 units, but the size will vary. And cytoplasmic movement is very rare or absent, because no cell organelle in it. But here in eukaryotes we can see the cytoplasmic movements can be seen, because they need to circulate all energies to all the cell organelle. So cytoplasmic movements can be seen in eukaryotic, but it is absent in prokaryotes. And vacuoles are absent in prokaryote, but it is present. And lysosome also absent in prokaryotes, but present. Lysosome is a organelle. And growth. And cell can grow in both aerobic and anaerobic condition, and in prokaryotes, but eukaryotes they need aerobic condition, and the cell sizes ranges from 1 to 10 micrometer, but here 10 to 100 micrometer. Is that clear, children? So that's all about the difference between prokaryotes and eukaryotes. And next class we'll start one example for prokaryotes and one example for eukaryotes. Is that clear, children? So next video I'll send the structures and organelles. Okay, thank you, ma.